يعني هيفتح قدامكم شويه، وهيديكوا ليكتشر عن الانترودكشن اوف الفارماكوجينومكس، وبعد كده ان شاء الله باذن الله انا هديكم يمكن 3 أو 4 محاضرات ورا بعض. اعرفكم بنفسي، اظن ان في كتير مش من الكليه اصلا. انا دكتور إمكا، الكمبيوتر بتاعه اتصلح واضح.
Yeah, it just froze and it's not responding at all. So I'm trying to log into another laptop. Okay, so to see.
Okay, take your time. I will introduce myself to them until you manage the problem.
Oh, okay, just opening now.
Okay. So هعرفكم بنفسي. دكتورة نانسي صفوت، PhD Pharmacology, Pharmacotherapy Specialist. إن شاء الله زي ما قلت هديكم جزء Pharmacogenomics مع دكتور إمكا. بس كده. حد عنده أي أسئلة تانية؟ حد عنده أي أسئلة؟ لسه مفيش حاجة، صح؟ مفيش أسئلة لسه. طيب كنت عاوزة أعرف إذا كان حد منكم دكاترة خد كورس الفارماكوجينومكس في الأندر جراديويت، يعني في دراساته قبل كده. يعني عاوزة أعرف هل عندكم باك جراوند ولا لأ عنه. طيب دعاء، في حد خد دعاء؟ روابي انت خدتيه معانا يا روابي صح؟ أسماء، اوكي. طب كويس، في ناس خدته، في ناس عندها باك جراوند اكسلنت. خدوه عندنا في الكلية. سارة، اوكي يا سارة. ماشي، سارة وروابي، تمام.
Yes, I think
انت خدتي الناحية الثانية يا رزان، خدتي البايوتكنولوجي. Yes, Dr. Emka. I was just asking them if any has took the course before, if any has a background on pharmacogenomics before or not.
Okay, so yeah, good afternoon or good evening, everybody. Just start to record this section. Yes, the technology is always failing, and we know that. So, I don't know why this system, it's a new system, is not opening my USB, because the course plan, which I'm going to post on the blackboard, and maybe send it to your class leader. The revised one, where I, put the topics as I'm going to explain. So this is the only one there that I need to open. So we can't just waste time, and let's just continue. I hope everybody is hearing me loud and clear. Yeah. Hello. Is everybody hearing me?
Yes, doctor, yes. They can hear you.
Yeah, okay. I can see some names here, like Mahdi Bujbara. Is it the same Mahdi? Yeah, for avoidance of doubt, your mic is open. So it's going to be interactive, and we need to speak, to each other. Okay, we need to communicate. We need to speak to each other. So you speak to me, and speak to me and Dr. Nancy, and then we can we have a discussion. Okay, because it is a master's program, and we expect that most of you are mature students, you have experience, and so our own is actually our role is to guide you in the learning. That's our role. So all the information that we need or you need in this course is out there in the open. Okay, so we will get some information, within the time frame of the lecture. We will present it. Okay, but you know that everything will not be in that lecture except through discussion. So we're going to have synopsis, and then we can go in details depending on how the discussion is going. So that is how it's going to go. So this is Pharmacogenomics MSc class, and I'm going to be anchoring this course with Dr. Nancy. We welcome you into this lecture, and the aim of the course is that at the end you'll be able to critically evaluate, at independent evaluation by everybody, and be able to describe and explain genetic basis for disease, individual differences that occurred in drug metabolizing enzymes and transporters. We are all familiar with this. To be able to explain the impact of drug disposition and action that underpin the practice of personalized medicine, because that is the goal, where it is going, personalized medicine. And be able to explain the application of whether single nucleotide polymorphism or polymorphism itself, and other genetic variants or variations, and their potential impact in clinical application, to patients. So what we are saying is that it should be, at the end of it all, clinical evaluation of the genetic variations will help in drug selection, drug dosing, and avoidance of adverse effects, and of course, what we call a regimen optimization. Most of you are familiar with what I'm saying. So we're going to, of course, use the Blackboard, this online lecture, thanks to COVID-19, and but it would have been, awesome for us to meet in the gathering and be able to, have a rapport. So we should be able to, in this regard, then we should be able to carry out interactive learning. We should be able to discuss. We should have some. We're going to have some case-based scenarios to use for application to follow up. So basically, the whole thing is underpinning on the course objective, which includes the description of the concept and the fundamental nature or concept of pharmacogenomics principles. First of all, defining the basic genetic and genomic consequences, concept of drug use and variations. So we should be able to explain the impact of drug disposition and its action that underpin the practice of personalized medicine. Okay, so just these are the objectives, and these objectives are reflected in the topics we're going to give you over this period. Okay. Now I've selected some textbooks. So luckily, some of this first textbook we're going to put it online. Dr. Nancy has have electronic copy, so we're going to put it online. But there are the other materials you can get. There are some of them in the library, which you can also lay your hands on, and we'll be able to use these resources. And I know that some of these resources are available on the internet, so you know how to do that. Other learning materials that we have put here, just for example. So when you click this when you click these links, it can take you to other materials, other learning materials. There are numerous, much of them on the internet. Okay, so you can avail yourself of their use. Yes, what we are doing here now, we are looking at the weekly course activity. How we're going to, be presenting this course to you from time to time. So now we are here giving you a brief introduction of the course and the expectations. So we just defined the objective of the course, the outline of the course outcome. We're going to talk about show you the assessment method, so that you know how this course is going to be assessed. The mode of course delivery, we've just talked about them. We're going to go through Blackboard, and yes, sometimes we will, as time goes on, we begin to look at other modalities like assignments, like presentation. We're going to talk about that later, and then your responsibility in being part of the whole course plan is important. Okay, so I will crave your indulgence that you find time to participate and make it an exciting period, so that we can learn, exchange information, discuss, bring some ideas on board, and some cases, and we'll be able to explain and share the information. So essentially what it is information sharing, and then we can guide you on the information, on the proper way to use this information, and how we're going to evaluate this information for your own benefit. So yes, time permits, we're going to go through a brief introduction from me, and just to remind us all, because if we haven't had this background before, then it will be an opener. But I know that the curriculum of PharmD of pharmacy, mostly you must have come across pharmacogenomics, pharmacogenetics in across the board somehow. We know that. The next one we're going to talk about, we're going to swap this. Actually, I'm going to send you the updated one because it's not opening here. The concepts of pharmacogenomics, and then we'll go to the principles of drug action and body transformation. It's a link between the two of them. So Dr. Nancy is going to take that. Fundamentals of pharmacogenomics, drug exposure, and the clinical response variability. So sometimes you will find out that there will be an overlap. Okay, there will be an overlap in topics, and you will see that, because we'll use that overlap to explain the core reason, and the main ingredient of that topic. Yes, we're going to also talk about genetic basis for disease and their individual differences. We're going to talk about the risk for adverse events and the avoidance of those risk adverse events. Genotype, specific dosing, and gene disposition. Mechanism of drug action and polymorphic drug targets. Okay, these are some of the topics you can find them. Clinical pharmacogenomics, testing and interpretation. How do we test it? How do we interpret the results in the lab? Bedside interpretation. Everything is a patient care bedside. Clinical implementation of the pharmacogenomics in different healthcare settings. So we're going to give you such scenarios, and then see how it goes. Then there's maybe we're going to have an assignment. Some assignment like that, we're going to have that. And if you look at on this side, on the side of the course plan, we say homework, assignments, and projects. we're not talking about projects, we're talking about topics that will help us to understand the course. Okay, each of the topic that we're doing. Okay, so this will represent, as we go on, the plan will crystallize into something, that we will follow and be able to understand. The presentation on the case-based scenarios, on examples of hypersensitivity reactions that is occasioned by pharmacogenetics or drug response. The same case-based scenarios in treatment of pain, same case-based scenario in cardiology, oncology, and psychiatry as we go on. So we are going to develop it, and then we will see how this will interplay with our with the course delivery. So yes, as we know, you are going to be responsible for some of the materials. Like, for example, we ask you to do a search or do a presentation. You of course, you know that you're responsible for such materials, okay? And the library assignment, we're still going to decide on it and see what's going to happen. Whether we're going to do an assignment or whether we're going to do a presentation. But I would prefer a presentation, okay, with topics, and this will be part of a 10% of the total exam, as you will see. The examination and assessment will be as follows, okay? So we will announce quizzes. Within the topics that we covered, we will announce quizzes, and these quizzes will include topics that are covered from the lecture before the quiz, and they should be 10% in all of the final grade of the course, okay? So we're going to have also two other examinations, the midterm and the final exam, and the outcome will be about 30% and 50% in all. If you add the presentation into the mix, you will have 100%, and that's how we displayed it here to show you how the assessment is going to go. Yes, so we expect everybody to be part of it, to attend. Because if it's a Blackboard lecture, the Blackboard will generate the attendance and the participation. So, and the Blackboard will give us the attendance, irrespective of the attendance we mark on the banner. So, let us try and be part of the presentations. So, if you have any questions, if you have any anything to say, anything to add, it's a discussion. It is an interactive. It should be an interactive lecture. It should not be just one-sided. Okay, from me to you. Okay, from Dr. Nancy to you. But it's better if we have a discussion. We have we prime ourselves in that nature, and then we can guide ourselves in the direction we go. Most of the time, the discussion in the class will be a part of the exams. Most of the time, the discussion in the class will be part of the exam, because what we are going to discuss will be what is related to the course. And so when we ask the question, we will discuss, and eventually maybe we'll ask the question in exam. It is what you expected to know. So that will cover part of the intended learning outcomes of the course. So we're not going to go outside the course intended learning outcomes. Okay? So if you have any questions, anything how you want us to modify or carry this lecture along, so that everybody will be carried along. So you can give that suggestion now. The mic is open, so everybody should be free to say something. Yes, are you people still there?
Everything is clear. Thank you, Doctor.
Yeah, Abdulaziz, yes. Yes.
I want to ask about any reference there or books can study from there.
Yeah. Okay, the reference books. Yeah, in the course plan that I'm going to send to you, put a reference material, a reference book there. Like I said, we're going to give you an electronic copy on put it on Blackboard for you to avail yourself of the use. Okay, but there are loads of textbooks with the topics with different emphasis and focus and explanation that also help to understand. So it is not one mode fits all. You know, some might just select, talk about polymorphism, okay, genetic polymorphism. Some might just talk about personalized medicine and how it affects the patient bedside. You know, these are information that are there on the net. So, like I said, we will try to get this information, present them in synopsis, and then we'll have a discussion with them. Okay, have case discussions, case-based scenarios, and so that for better understanding and application. Okay, because in the exam we're not only going to ask to be able to achieve the ILOs of the course. We're going to ask questions based on knowledge, based on cognitive and application. So that's where the case-based questions and scenarios come in. Okay? Yes, any more questions? Yeah? No more question? Okay. So, in absence of any other question, Dr. Nancy, do you have anything to add to this?
No, Dr. Emka, no. Okay, like Dr. Emka said, it's an interactive. So if you have any questions, any doubts, just ask. Okay. Probably next lectures I will be presenting the pharmacogenomics concept. So I will send you a paper. Please read it before our next lecture. I will send you a paper which introduces you a little bit about pharmacogenomics, the expressions we use a lot in pharmacogenomics. Please read it, and if you have any questions, prepare it for the lecture. I will send you also a video. Please watch it, and we will discuss it during the lecture. Okay? Is it clear for everyone? Okay.
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 So,
thank you.
Okay. So,
thank you.
Okay. Thank you, Dr. Nancy. I will go now to do the introduction, just an overview of what to expect, generally what to we'll see likely be sharing with you in the different lectures. Okay. So let's okay. Okay, so we're just going to go into the introduction. I don't want to take so much time. Like Dr. Nancy said, yes, from time to time, reading materials, and we have there's a lot of reading materials. Okay, what do we expect in this regard? Okay, So first of all, in this introduction, we have 4 learning outcomes. That is what we need to take out from this introductory lecture. Okay, like I said, it's an overview of what we are going to be doing. Okay, definition of pharmacogenomics and understanding terminologies associated with each knowledge. So just to refresh you, because I know some of you have the background already. Brief historical background of pharmacogenomics, how it is, because this is coming from a pharmacological point of view. This describing the basic principles of genetic inheritance. What are those things? I'm not going into details here, just to say the high points that will later be discussed in details in each topic. And then be able to cognize the concept of individual differences relating to drug therapy or drug response. Okay? So, first of all, when we talk about pharmacogenomics, it's just a 2-word, like we all usually say in pharmacology, 2 words joined together. Pharmacogenomics, the genetic genotypes. So it's you have to the definition of pharma and genomics comes together. That's what gives you this gives the complete picture. But on the part of the genomics, it is a study of the complete set of genetic information in a cell, in an organism, a human individual. Okay, and that goes into what we call terminologies like genetic polymorphism, multiple differences of gene sequencing found in at least in a population where we say yes, this thing exists. In the case of different alleles that are found in a loci of a gene. Genetics. Basically, the study of heredity and variations. Now, if you join this together with pharma, so it tells you the story. That is the story of pharmacogenetics, pharmacogenomics, and things like that. Okay, that's where we're going. Okay. Now, what we just said here, we just talked about the different types of the genetic sort of heredity and its variations. We talk about the genetic polymorphism, okay, multiple gene differences. We talk about the SMP, which we'll talk about a lot. Single nucleotide polymorphism. We'll explain what that means, in terms of gene, genetic variation of a single gene in individual. Okay, so we have the entire knowledge. Having defined genomics and genetics, genetic polymorphism, so we come to pharmacogenomics and we say it integrates the knowledge of pharmacology and genomics together. It's an integration. Okay, it is a relationship between the genetic variations and the response to drugs. By having this genetic understanding and information, so there's a potential to increase drug efficacy and reduce toxicity or side effects. That's sometimes that we see in patients that might be idiosyncratic in origin. So taken together, we can say it is the story of effects of genetic differences among people and the impact that these differences have on absorption, efficacy, metabolism of drugs, and toxicity. If you look at this, these two, for absorption, efficacy, metabolism, and drug toxicity, encompasses pharmacokinetics and pharmacodynamics together. So, but this is a breakdown. Okay? So we can say impact of differences that have on pharmacokinetic or pharmacodynamic of drugs. in a patient. Now, the other aspect, the difference between when we talk about pharmacogenomics and pharmacogenetics, sometimes people erroneously use them interchangeably, which is not true, because this is a study of influence of individual factors that predispose a patient to respond to a drug, okay, of a gene. But we talk about genomics, it has to encompass we're talking about the genome, we're talking about the entire, particularly we talk about in a population. Now, the origin of this has always, the origin of this background is from, we know the famous mathematician Pythagoras, that is, recorded the first inter-individual difference, the drug administration. This was in 500 BC in some patients who developed a hemolytic anemia after eating a beans. So we know what this means. This favor, which we have come to know that this is a G6PD patients, okay? But it was not defined then. But he noticed that, okay? So the term pharmacogenetics was first used and coined by Vogel in 1959, okay? And it had to wait until 1962 before the study of hereditary and the response to drugs was finally defined and come into focus by Carlo. And then, like before then, in the 50s, that was when it was discovered. Before this was happening, they discovered the abdominal response of succinylcholine, okay, to butyrylcholinesterase in patients, okay. One, because they lack the enzyme, and so there is toxicity. That's what we call apnea, okay, respiratory apnea in those patients because of extensive neuromuscular blockade. So it affected them, they have that apnea. And then also, they found that the development of hemolysis in malaria patients who were deficient for this G6PD when they were given primaquine. So all this led to the definition later by Carlo to define this incident, and this incident was found to be increasing in the population, and so that is how this came into being. Now, the genetic inheritance and gene expression help to throw more light on pharmacogenomics and pharmacogenetics principles. So the characteristics were determined by genes, or units of genes are called units of inheritance anyway, which are determine the characteristics displayed by every individual. So the particular form of gene that appears in the locus is an allele. It might be responsible for hair color, it might be responsible for eye color. Okay, so these are some of the things that we are noted. An individual inherited a pair of genes, one from each parent. But according to the person we call the father of hereditary study, Mendel, okay, Mendel in his all his researches was opined that when somebody inherits from parents, that there is no mixture. Okay, there's no creation of mixture. It's either you express one or you don't express the other. Okay, and that gives rise to what we call the dominant and recessive. Okay, but sometimes I'll show you that sometimes in nature, depending on the genetic makeup, you see the phenotypic expression will change. When allele is dominant, it is present. When it is recessive, okay, it's not displayed, but it depends on the composition as you are going to see, okay? Both alleles could be passed on to offerings without being altered in the process. So it will not be altered, it will just be there present. But somewhere along the line, this position, along the line of generations, it might get expressed, like that. So if a recessive form was not displayed in one generation, it could be displayed unaltered in subsequent generations. That is how this happens. Some traits are determined by the combined effect of more than one pair of genes, and that is why we say they are polygenic. When some traits are polygenic, so we say they are polygenic. I will give you an example of polygenic. I'll give you an example later. Now, if you look at this, because in gene expression, we have the genotype, we have the phenotype. Okay, the genetic makeup of an individual contains a matched pair of alleles for each of the traits that they have. Now, look at an example of this. What I was telling you, each allele of the matched pair exists independently and as a discrete unit, whether it is dominant or whether it is recessive, because recessive is not expressed when there's a dominant. So if you look at this picture, it has a dominant allele AA. So this is the color. Now, when the A and the recessive are present, it changes to pink color. So, and this is recessive. This is homologous. This is heterozygote, and this is homozygote. So they are. You can see the difference of expression. This is just how Mendel started this study. Now, the phenotype is just the outward appearance and other observable characteristics of the individuals, like we said, such as white flower color when both alleles are recessive, or purple flowers when they are both, or both the allele is dominant. When it's a dominant, like I just showed you, okay, you see the colors are shown depending on the whether they are homozygote or heterozygote and things like that. Okay. Now there are other things that we'll see that will affect the gene or affect the responses or genetic variation. And the ultimate, what we're saying is that the genetic variation will ultimately affect the drug responses. And that is why we're talking about pharmacogenomics or pharmacogenetics. Okay? There's what we call the modifying genes in individuals. Okay? They alter how certain other genes are expressed in a phenotype. For instance, there's a dominant cataract gene, which will produce varying degrees of visual impairment. So it will produce various, so that means it modifies other genes in the body, as we will see. There are others that are called regulator genes. These regulator genes can either initiate or block expression of other genes, and they control the production of a variety of chemicals in the body. Then you have the pleiotropy. The pleiotropy. This pleiotropy is usually what you see as a single gene that is responsible for a variety of threats within the body. For example, the sickle cell anemia or sickle cell trait. This gene is not only causes the sickling of the red blood cells, it also affects the heart, the kidney, and causes a lot of eye problems. So that's why they call pleiotropy. So these are some of the things that you see how a gene can affect, cause other diseases. So this, Dr. Nancy will elaborate more when he talks about genetic diseases in the subsequent topics we're going to talk about. So this just to give you an idea of what we're going to be talking about. Now, there's what we call the stuttering alleles. Now, if you look at these stuttering alleles, like we said, when a recessive gene is in an individual or from parents, now over the years, it might not express. It can express according to a later in the generation. Again, this is different in that one allele in the gene that causes severe symptoms of a particular disease, when it passes to other generations, it multiplies. And as it multiplies, it becomes, the severity of the symptoms increases in those offerings. That's why we say that more severe symptoms in each succeeding generation due to the segments of the defective gene being doubled in their transmission over the generation. So you find this in individuals. Now, if we look at what we call the defining individual responses to drug therapy, because that's when we take the genetic variations, different gene polymorphism or single nucleotide polymorphism, we take them together, okay? We now put them on the same board and see how the individual with such variations respond to drug therapy. Okay, and that is where we're going. And in the sense, we will now look at way to mitigate. How do we mitigate this? Because that is what it is: personalized medicine, bedside treatment. Okay, for that single patient. So for that response, okay, like we always say, you give the right drug to the right patient at the right dose at the right time. Okay, that's always same. So, but you find out that prescribers' knowledge of appropriate drug, when it comes to the people who are who have the genetic variations, okay, they are always skewed towards normal average dose for average person. Okay, so because their data derived from individuals, so because if you look at those people in a society, in the population, maybe they are about 1%, maybe they are about 2%, maybe they are about 5%. But the majority of people are normal responders. And so the likelihood of treating everybody as normal responders is there, is innate in the practice. So with actual dose response relationship for particular patients is not certain, because you don't know until you find out what is the makeup that is affecting this response. Okay? Like the treats, they can never be certain that their choice will be enough. Therefore, the effectiveness or the safety that they must recognize, they have the need to monitor the outcome of these patients if they don't know how the patient is responding to these drugs. So individual variations from normal is often predictable. And like we said, some very good prescribers who are able to anticipate and adjust, but not in patients with genetic variations, either in disease presentation, either in drug response. So you will see that. Okay, and that's why this is the benefit of this course, to be able to understand that and be able to have an intervention in to such patients. Now, the altered response. That is, we're talking about individual response, variations due to the individual response. So the variation in drug concentration, now we're talking about pharmacokinetics, is achieved by equivalent doses. Okay? And it's much more important. It's much more important cause of this individual variation that we talk about, that they counter in patients' care. because the same dose might produce therapeutic effect. The same dose might cause toxicity in the same patient. So this is what we'll be looking at. So, and there are so many reasons where you can say, you look at it and say, this might be the cause, it might be due to maybe absorption, maybe due to lack of distribution or more distribution or extensive distribution or metabolism or excretion of drugs, maybe due to the body volume of the patient, because we have certified dose already. So and this is what you can encounter. Yes, fast metabolism. Okay, you see that in a patient. So you see some people, some people are, they use the term, there are 3 terminologies. Okay, we call it the ultra fast responders, metabolizers. We have the extensive, which is called as the normal responders. They don't have the poor metabolizers. That is, either they are lacking the enzyme or they don't have the enzyme. So these are the 3 categories, okay, which we will expand when we come to drug metabolism, okay. The variations may be also influenced by what we call the genetic polymorphism, which we have described, and it can result to the altered responsiveness in drug targets. The first one we talk about metabolism, absorption, distribution. We're talking about kinetic profile that can affect that, which we're going to expand more. Now, in this altered responses in drug targets and altered metabolism by enzymes activity. So we are looking at the pharmacodynamic effects, okay, and see how this might affect, because we will just, that's what Dr. Nancy will take up on the, maybe the next 2 lectures. Hence, the nature of the relationship between genetics and the response to a specific drug, once it's defined, okay, once it's defined, then the physician or the pharmacist will be at home to be able to reduce the emergence of toxicity and maybe have a good outcome. So that is what we're talking about. Those who suffer adverse effect becomes evident. You see them. You know that this patient will suffer, and then you'll be able to mitigate the effect of that patient. Okay. So we have various examples to give, which we will give individual when we come to those topics that we're going to talk about. Okay. So in short, the focus will be generally to understand the mechanism of clinically significant drug interaction that will predispose a patient, to either respond or not respond, either to be one of the 3 categories we've just mentioned: ultra, normal, poor responders. Should be able to recognize potentially clinically significant genetic influences, either in the disease, like I showed you. Some people can exhibit more severity of symptoms than the others on drug efficacy and toxicity. Okay, drug efficacy and toxicity. For example, we're talking about significant genetic influences, like those with GCSPD, like those that lack the alcohol, the hydrogenase enzyme that will break down alcohol to aldehyde and acetic acid. So if they lack that enzyme, they will have the anti-abuse kind of reaction. So that's what we're talking about. Genetic predisposition to diseases, because once you have a predisposition to a disease due to the genetic variation or the presence of an allele or a gene that will be able to dispose that patient to this disease, okay, that will be able to affect either the disease progression, the drug that the patient is taking, or even the transport of the drug, within the body, the distribution. So we'll be able to describe polymorphism types and their impacts on kinetics and dynamics. Also, importantly, because in the US now, this genomics is part of the pharmacy curriculum, okay, both in PharmD and in the master's level. And so because they are going into that, a lot of adverse drug reactions from the genetic variations. So you need to understand the ethical, the legal, and social and economic issues related to this pharmacogenomic testing. It's important and it's increasing in the US. Also, we hope that you identify resources that are current, to update pharmacogenomic information, because that is where it's to go now. The ACPE, they're laying emphasis on this. The CCAP, they're laying emphasis on this. So it's an important area to go to. Now, after all these are tending towards what we call the personalized medicine. Because if an individual, the genome, the entire human genome project was in 2004, I believe. Yes. Somebody correct me. It was 2004. Okay. And since about 95%, so the human gene. And so you can be able to identify those individuals through genetic testing and know their kind of, predict their kind of response and be able to personalize the drug that you give them or their treatment. So the move towards personalized medicine is based on genetic testing, and it has the potential to improve, that's what it is mostly, the risk to benefit ratio of drug treatment. So you can imagine, for example, most of you who have read clinical trials reports, or sometimes in doing clinical trials, it fails. One, because they are not, they select the patient, the volunteers randomly, and so when they select them, randomly, you get such scenarios, such effect. Now with genomics now, it will even help the drug companies to be able to clinically try their drugs on the specialized patients. They know that these patients have this and this drug has this, so they will have a better outcome. So because many drugs are lost in development because they cause an unacceptable rate of the adverse effects, in selected patients, So if with genomics, you'll be able to know your patient or your category of people you're testing the drug for, and it will even help the pharmaceutical company to even reduce costs of this testing. Because any testing that goes clinical trials and there's an adverse event, a lot of money go into that. So that is what it takes. So generally, this is what we're going to expect in this course. So I expect this course to be an exciting one. I expect it to be, for us to go into it. It's a different kind of new well, I can say it's a new area, okay, where people should come in because the medicine now is becoming personalized. It's no longer one blanket, covers all. So it's for patient outcome, better patient outcome, and also you can imagine a place like UK or US. When you give a patient a drug like that, okay, and the patient has comes up with any adverse reaction or adverse effects or even mortality, your license is on hold. All the people that are involved in that drug administration for that patient, their license is on hold. So because you are expected, to be able to have told the patient or have information on that patient before giving that medication. So people are very careful now, more careful, because they can lose their license for things like this. So that's why this is expounding. Okay. So this is a brief introduction. So we're going on all these areas. We're going to expound them in each topic. So just to sensitize you. Okay. So if you have any question, interaction, yes, you can ask. Hello, is anybody in the house? Okay, so please, when next I'll be on this platform, I want us to interact. Okay, so we're going to make sure that you do that. So please, this you don't have any question, and we didn't take any break. So Dr. Pharmacogenomics is broader than pharmacogenetics. Yes, pharmacogenomics. Yes, if you talk about pharmacogenomics, it has a broad application than pharmacogenetics. Okay, so we are talking about in pharmacogenetics. We talk about mostly about SNPs. Okay, the difference between phenotypes and genotype. Now, the genotype is in types of the gene. Okay, it's a type of the gene. Okay, when you talk about the phenotypic expression, is outward expression that you see in a patient. I mean, in an individual. Okay, so it might be the color of the hair, might be the phenotypic expression, it might be the color of the eye, okay? But the genetic makeup, genotype is different, within that individual. So what is expressing, because is gotten this information from both parents, and then depending on which one of them is either if it is a heterozygote or AA or A, it will express just like what I showed you. Okay, the genotype. If you look at it, let me go back to that picture where I gave you the AA and the homozygote and the heterozygotes, and you see the difference. This picture here. This picture here. Okay, these are the expressions. These are expressions. These are the makeup of these expressions. So you have the dominant here, you have the dominant and the recessive, and you have the recessive here. Okay. Razan, is this the same Razan that graduated from here? Okay, that's interesting. Yes, 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 yes. I remember you. Madi, are you the same Madi that graduated from here? Oh, Madi Bujbara. Okay, that's interesting. Okay, so we know each other. So I think that's it. So the next class will be with Dr. Nancy. I think she has I don't know whether she's left. Okay. So yeah, she will have the next class with you. So I thank her for assisting me in this course. So thank you, everyone. I will post the, I don't know, Sarah, I think I will send the something to you. I will send the course plan to you, and I'll put it on the blackboard. So for those of you who are on my blackboard, so you can, if you get anything on the blackboard, you can share it with those with Dr. Nancy. Okay, Dr. Nancy's blackboard, because they are not yet connected. Because if they are connected, then I will send to you, I will send to her, and she will, and I put it on my blackboard, everybody will see it. Okay? So please, if you have any questions, anything, don't hesitate to ask us, and we'll see. Okay, that's good. Okay, that's good. Thank you. So see you after Dr. Nancy, then we'll come back again. Okay, bye. Stop recording.
طيب دكاترة، كده الدكتور إيميكا خلص إن شاء الله بإذن الله. أنا محاضرتي معاكم من الأسبوع الجاي إن شاء الله بإذن الله. زي ما قلت لحضراتكم هبعت لكم بيبر هيبقى فيها جزء كبير من المحاضرة، فيا ريت تقروها قبل ما تخشوا معايا المحاضرة. وكمان هبعت لكم فيديو يا ريت تتفرجوا عليه، بحيث ان انتوا داخلين يبقى عندكم اوريدي باك جراوند قبل ما ابدا اتكلم، عشان الموضوع ما يبقاش بالنسبه لكم كله جديد. تمام كده؟ اشوفكم الاسبوع الجاي ان شاء الله باذن الله في نفس الميعاد الساعه 4 الساعه 5 سوري الساعه 5. يلا السلام عليكم.